Okay, good morning, ladies and gentlemen. My name is Intisar. One of the co-presenters this morning. Fine. So your first presenter, he is Mr. Ian Burrell. And just to present his biography, he was raised on the South Shore of Montreal, pursued his undergraduate and graduate studies in bioresource engineering, focused on food engineering. He had a particular focus on the brewing industry. Studying on campus, he also apparently attended beer school twice. Once to become a brewer and then to become a sommelier. He's also a judge for competitive beers. He's excited to finally be pursuing PhD in brewing science and fermentation technology under Dr. Horsett. So, without further ado, please welcome.
All right, thank you for that introduction. Appreciate it. I want to start the presentation off with a quote that says, "Co-culturing techniques are of great importance in synthetic biology and are widely seen to hold enormous potential for foundational research as well as novel industrial, medical, and environmental applications." And that's taken from the paper, "Co-culture Systems and Technologies, Taking Synthetic Biology to the Next Level." So I chose this quote and highlighted the reasons why I wanted to talk about co-culturing. It's novel, it's great for research and development, so great to present to a room full of grad students. It has novel potential applications in industrial and environmental. So co-culturing is really quite simple in concept, but only in concept. It's using two or more types of cells in the same medium. They can be cells of different strains, even different phylums, or even different kingdoms. It doesn't matter. But for the rest of this presentation, I'm only going to talk about co-culturing yeasts together. To take advantage of different traits, you can mix different yeasts together and get all these benefits at the same time. That's a lot easier than having a genetically engineered strain that would have all of those traits, as well as being a lot less expensive than having it engineered. Sometimes it's just impossible to add certain traits to certain strains. There are also some issues. It's very complex, much more complex than single cultures, and it's still being studied today, and it's still kind of understood or misunderstood. It takes a lot of knowledge to control it in terms of quality, so you have to hire more specialized personnel, and then of course pay them more. And there's a loss of product consistency over the generations, meaning you need a new inoculum at the beginning of every batch. And finally, the question I'm sure you've all been wondering is, what the heck is killer yeast, right? Well, killer yeast don't kill people, they kill other yeast. And in fact, they don't even kill all yeast, they only kill specific classifications of yeast, so specific strains. Typically, they're classified into three groups. There's the killer, which secretes a toxin while remaining immune to the toxin itself. There's the neutral, which is immune to the toxin but does not produce any. And lastly, there's the susceptible, to which the toxin is lethal. And I wanted to point out, especially to graduate students, that the mechanisms of immunity completely unknown, and scientists have been chasing this for decades. Anyone who can figure out how they manage to remain immune while killing off all the other yeast is pretty interesting. So probably a Nobel Prize in there for somebody. There we go. So a couple examples. Champagne yeast and wine yeast tend to be killer yeasts, and brewing yeast tend to be susceptible yeasts. There are neutral yeasts in both categories, and there can be killer yeasts in categories. But just to give you an idea, wine and champagne tends to be a killer strain, whereas beer tends not to be. So with that in mind, neutral yeast can be combined with neutral yeast or with susceptible yeast, and everything will go quite well. They get along, they're immune to the toxins, while the neutral is, and they don't produce any. However, they can also live with killer yeast. So neutral and killer go together, and killer go together, but they're probably not as happy about it since they will be producing the toxin. So not exactly peace and love, more grumpy cat, but still functional. None of the yeast dies. But if you combine killer and susceptible together, the susceptible will be completely eliminated, which can be really bad if you want to, say, to have brewing yeast, but it can also be really good, right? So it can be good or bad news, and it can be good news because of the case of wine. When you make wine or champagne, unlike beer, you can't boil it, right, or pasteurize it even. You're going to lose a lot of flavor. You're going to create pectin haze. and you're not going to have the aroma you wanted. So when you collect all your grapes out of the field, they're loaded with bacteria and yeast on the surface. So you crush them, and right now it would go spoil really, really quickly. But when you add your own inoculum of killer yeast, like wine yeast, it'll kill off all the wild yeast. But what about the bacteria? Well, they die because of the alcohol, the low pH, and the lack of oxygen. So you actually don't even need to sterilize your grapes or sanitize in any way. You can just add a good yeast starter of wine yeast because they have the killer gene in them. In fact, there are 2 genes. And lastly, I found it was way too interesting not to talk about how the toxin is produced. It's not produced by the yeast itself, but instead by a virus that lives inside the cell of the yeast, which is really interesting. It's an icosahedral double-stranded RNA virus, right? So icosahedral just means it has 20 sides. It has nothing to latch onto a cell membrane, and it doesn't reproduce. So it produces this protein, it gets cleaved by an enzyme and secreted into the medium, where it is toxic. But the virus itself stays inside the cell. Doesn't replicate, doesn't kill the cell, and doesn't attempt to take over other cells. Because of that, it can't even be considered a virus. So it's considered a virus-like particle, or VLP. So already the viruses are hard to differentiate between living and non-living. Now we have a virus, we can't even differentiate between virus or not. So it's a very odd kind of system. In conclusion, co-cultures are extremely complex, and I'm a little worried about this question period because they're so difficult. Killer yeasts are both good and bad, depending on the utilization, and mostly the mechanism for producing the toxin is fascinating. Thank you very much to Dr. Dumont, Dr. Orsat, Christopher, primary secondary reviewers, the audience, and the university.
Okay, thank you, Ryan, for your presentation. It's just a continuation. So does anybody have any questions? Sure hope you do.
Just on that last virus, or the virus-like particle part, are you saying that's always present in the yeast you're talking about?
Yeah, so they don't even replicate except for one exception, which is when the cell divides. Well, we call budding in yeast. When it buds, it'll transfer a couple viruses to the next cell. That's it. Otherwise, they don't replicate or do anything else. It's kind of like a symbiotic relationship with a non-living thing. So I guess you can't call it symbiotic. But...
Next question.
Let's say you're brewing beer and you want to eliminate one kind of wild yeast there. Could you use a killer yeast while not killing the other kind of yeast?
Yeah, so sometimes what they'll do is they'll actually separate the 2 yeasts into 2 different containers, let them ferment through, and then mix them back together at different ratios or at different times in an attempt to kill the other yeast or maintain your own yeast. You can go at different times in the life cycle. So it's kind of an art at that point. You really have to go a little bit of trial and error, go to the math, and then you can combine them at the right moments to do that.
What would be the incentive to genetically engineer these things if it's complicated and expensive? Like, are they actually genetically engineered?
Well, so right now, I guess I put a lot of emphasis on beer, but right now they use co-culturing to produce enzymes, to produce polymers, to produce a lot of different food products like kimchi. Certain vegan styles are already co-cultured, and there's a lot of money to be made on that. I have something like, I wrote down for just the question period, 37 different examples of things produced by co-culturing. So there's platform chemicals, antimicrobial substances, microbial fuel cells, lots of stuff. So there's a lot of interest in doing it, but it's so expensive to genetically engineer it, they decide to do the same product with a cheaper alternative.
Okay, so that's it. Thank you very much.